open to other species that are vocalizing outside the context of flock. So as we are following a flock and you see another species that hasn't been part of a flock calling, the only way to distinguish is to actually speak into the recording as I am recording. So I'll say, okay, this call is not part of the flock. So that is the only way we could manage data collection. And simultaneously, I'll also note down the species that are part of the flock and the number of individuals that are part of the flock. So by following a flock, I have a list of participants, and the definition of a participant species for my study is very specific. Those species that have participated in the flock throughout the duration of my recording, because only then will I have a reference list of species that I can match to when I independently listen out their recordings that we have captured simultaneously. So we have a reference list of species, and we can convert this participant species into a matrix where each row here is a species and each column becomes a flock. So it is basically a matrix, a presence-absence matrix. Using this method, we were able to track changes in species composition and extract morphological traits from an open database, which is WAYANAD. And we recorded all these flocks using a combination of Sennheiser ME66 directional microphone and a Zoom H6 recorder. And how exactly do we sample such a huge amount of data? You're recording each flock for like 15 minutes. You have to track down the number of species within each time frame. So what we did was to subsample the recording. So I've binned it into 1 minute time frames and consider this as a 7-minute recording. So I will be sampling every other minute to ensure that overall I get a sense of the entire recording and I evenly sample the recording throughout its entirety. And I would census for different participant species that I could hear through the recordings, and this was mostly done based on the visualizations provided by Raven Pro. And now we have another matrix. This is that of a vocalization matrix. For each time frame of recording, which species had vocalized during that time period. Now, what can we do with this information? And a straightaway answer is we can calculate the vocal activity index. We have the time frame, number of time frames in which a particular species had vocalized while being part of a flock. And we also have the total number of time frames in which the species was actually a participant of the flock. So a basic division would give us a probability of that species vocalizing when it is being part of a flock. And this is the vocal activity index. And this was done using visualizations. The other parameters that I measured was using the spectrograms. A quick definition. Spectrograms are the visualizations that we get when we record a sample and put it in Raven Pro or other softwares that can deal with these recorders. And on the x-axis here, can you see it? Yeah, you can see it, right? I'm just confused. On the x-axis is the time lapse, and on the y-axis is the frequency, and the darker the shade is, the more amplitude that particular space has. So what parameters do we extract when we are dealing with a system like a flock? Here we should be particularly careful because most frequency related parameters are reliable, and but however amplitude based parameters are not reliable because your direction to the bird's direction, everything it would influence what the amplitude is, and therefore we generally rely on a set of frequency related parameters that when taken together, broadly the idea is to define the shape of the note that we are trying to characterize. So this is a multivariate data, right? It has multiple information across different axes. So using an ordination test, we are going to crunch it to a fewer dimensions, and running a PCA, we get information on where each individual notes lie on that space. And and that PC1, that one axis explained around 80% of the variation of these entire data that is collected and crunched. And we used the average PC scores for each species to denote their spectral signal space. So that is their acoustic space, if you want to call it loosely. So the analysis broadly follows like this. We have an observed data, which is the flock species composition. And then we have also independently gathered data on their phenotype. The two traits we are interested, one is the morphology related traits, body mass, which came from the open database, and the other is the acoustic database, which is the PC scores that we just spoke about. Now, we can replace the ones that is present in the matrix. Let's say a species A is present, we say it as 1. We can replace that with these phenotypic traits, and then we can create random flocks. What is a random flock? If we were to if we have a community of species and we were to randomly put species in together, what that trait space would look like as a community is a random flock here. So here, a simple way to do it is you have rows that are species, columns that are flocks, and you fix the total number of species and pool in the species based on their probability of occurrence in the entire matrix. So basically those are null flocks, and we can do it 1,000 times, and for each iteration we can calculate the standard deviation of these traits. So that is the variation of these traits, because we are trying to understand how similar these traits are. And this can be compared to what is expected by chance. So we have the 95 confidence interval from the 1,000 null flocks that we have generated, and we can compare that to what we observed, the composition we observed. Is that clear? That was a little complicated, but I hope that was not too much too. Yeah. So coming to the results, the broad result, as you might have already expected, is that the flocks were generally dominated by smaller sized bird species. And this could be a result because we are dealing in a system of moist deciduous to dry deciduous landscape, mostly composed of canopy species. So mostly smaller size species might be dominating. But in the system, what we see is that when we map this body size onto their phylogenetic tree, we see that the smaller size species are generally closely related compared to their larger counterparts. And we have independently also measured their vocal activity index, and when we superimpose that on a phylogenetic tree, what we see is that the smaller species are also the ones that are relatively more vocal when they are participating in flock. Also, to test the already established paradigm, are the species that we are also observing similar in body mass? And this is the basic framework. You have flock richness classes ranging from small flocks to large flocks, and most of the flocks fall within groups composed of species ranging from 2 to 10 species. And these constitute 87% of all the flocks that we have observed. And what we see is that the standard deviation, you focus on the orange dots here, that is what we have observed. The variation in the body mass is much less than what is expected by chance alone. What the expectation and the chance space is the whiskers that we see, the blue space. But our observed data falls below it, suggesting that more similar size species are indeed flocking together, which is the global pattern that we see. So that really applies to our system as well. Now what do we have? We have similar size species flocking together, and these are mostly composed of smaller size canopy species, which are also relatively more vocal. What does it mean? It means that there is a high risk of masking each other's information when all of them are calling together, which makes it interesting for us to explore whether there is acoustic similarity patterns similar to what we have observed with morphology. But when I did this with the PC scores, all the points across the flock sizes fall within random expectations. They are not extremely clumped like the body mass, as we had seen. So all the points fall within the expectations alone. Now, just to keep these two graphs side by side, you have a pattern in body mass where similar size species are grouping together, whereas in terms of their acoustic space, it's as if you're randomly pooling species. The variation in their acoustic is as if you are randomly drawing species and creating flocks. So this is where we see that there is difference in the patterns in both morphology and acoustic trait space in the mixed species that we observe in the WAYANAD landscape, despite the very strong correlation between them. And this is what we term as decoupling of these traits at the level of flocks. Now to just quickly summarize it, propensity to flock together with similar size species, pronounced in smaller size species. They are phylogenetically related, extremely vocal, but they are not acoustically as similar, and is as if they are random groups of species. And we have published this work this year, and please feel free to go through this work, and I'm happy to take any questions. You can scan the QR code. And what might be the way forward? All this that we have spoken about is generally called the cocktail party problem. How do you make yourself heard in a group when all of you guys are talking. So there are three strategies. You can use a different frequency range, you can call at a different time when the others are silent, or you can go up another floor and call from there. So I have explored one dimension of it, frequency ranges, and how overlapping frequency range might possibly cause masking interference or not in a flock system, but it will be more insightful to add the other dimension of temporal partitioning of vocal activity and frequency ranges as well. So with this, I will conclude my talk, and I'll have to specifically mention all the institutional support from NCBS, JNCSR, NCF, Preeti, Samira, and Anand, who has been extremely great guides for me. Manoj Kumar, my dad, Madhanan, who's my field collaborator, Arulka, Badrishka, NCBS Wildlife Community, family, friends, and all Kerala Forest Department too, and everyone who has supported me throughout this journey. Thank you.
Thank you. Questions?
Abhijit, excellent presentation.
Thank you.
It is not just the topic, but your body language, your the smile, the articulation, excellent. I think it was really one of the best for the this festival. Thank you for that, and also I'm sure that the flocking is so embedded into you that even your shirt has got a flock. So it's nice to see that.
Thank you.
When you showed that the flock, the different species, though I just recently moved to Wayanad, when I sit outside my house, I see the same pattern.
Right.
The racket-tailed drongo, the natachers, the tits. I don't know, you didn't mention the tits, I'm not sure about it.
Yeah.
But both the tits, the yellow tit, Indian yellow tit, the cinereous tit, all of them come together.
Right.
And also, I don't know if, but I don't find them every day. I don't see a pattern in them coming. Just once in a woodpecker, sorry, I missed the woodpeckers.
Right.
Rufous, black-naped, even pygmy woodpecker, brown-capped.
Yeah.
All of them come together. But I don't see a pattern. Is there a pattern of that, or timing or something that they come and go?
So if you encounter a huge flock at the outset, like composed of like 10, 12 species, they have already grown. So you will see such patterns in mostly in smaller size flocks as they start forming that flock. Like it's a very dynamic relation, right? A lot of species come in, move out. So that is why even if you look at the graphs, the larger flocks generally don't follow the patterns, because the priority there in some sense shift as you cross a particular threshold of the number of species. There you're trying to fill up a lot more niches, and there are a lot more smaller flocks are also coming together at that scale. So there picking up patterns might be a little challenging, but then they're filling a lot of feeding niches. That is the particular priority there. But here, as the flock forms, there the predation risk is more accentuated, and similar size species tend to start forming flocks. So
Yeah. Also, I had just one instance where I was sitting, suddenly I see a crested hawk, goshawk, coming and sitting on a tree. After 2 minutes, the first call come from the racket-tailed.
Right.
And the same species all flock together.
Right.
The same species which used to be seen together, they come at the warning call, all of them come around.
Right. All this data that I've shown you is outside an alarming context, because that is a completely different system. So as soon as there is a particular threat around, then literature has shown that there is convergence in their call in terms of the number of repetition of their call per unit time and the broad frequency ranges. So there is a convergence, but contact calls, this is the pattern that we generally see. Yes.
Good talk, Abhijit. One question: you have made a hard rule that a species is in the flock when it's throughout the period. Like that's what you've done.
Right.
Suppose you have to relax that to say 60%, 70%. Will that result change drastically? Have you tried it out? It doesn't look like. I mean, whatever flock we have seen more or less matches with what your conclusion is. But if you're done that, we clearly know that species come in and go back, and some of the species doesn't hold at all with the flock. They just come in, spend few minutes, benefit and then go back.
Yeah.
So if you lower the threshold, will your results change drastically? How is it?
I did that initially a bit, but then I didn't see a lot of species coming in and moving out. Most of the species, for the duration, we are not talking about half an hour, we are talking about 5 to 15 minutes. For that duration, it will take some time for us to even determine which species are part of the flock. By that time, that 5 minutes would have crossed. Then another 10 minutes we observe. Then so not a lot of additional species coming in and leaving in that short duration. And even if there, it's very few in number. So we've done a little bit of initial analysis based on that, but we realized it's extremely challenging because how do we acoustically sense this later on? We don't know if the bird was a participant when we are actually hearing it. It could well be a species calling from outside a flock. Yes.
Any other questions?
I was trying to understand the takeaway. So is there masking interference or not? Because they're all small, but the sounds are all at random. So that means there's no masking interference.
To a certain degree, we can just say there is level of masking interference as if you're randomly pooling on species. So they are not completely diverging in their call spectrum as well. And that is why it will be insightful to include a temporal partitioning of the calls as well when it happens in the foraging context.
Okay.
In an alarming context, the priority is not to communicate to each other as much. The priority is to get information. So even if there is calling at the same proximity which is observed, then it's fine. It's the alarm that matters.
When they're mobbing, they're all at the same time.
Exactly.
Right? Okay.
Yeah.
Hi, Abhijit.
Hello.
So, we are talking about flocking mostly on similar sized birds, right? So, I'm not sure whether it's a flocking or not. So, with respect to some water birds, I had seen stork, right? Stork sitting long time with cormorants inside an island near some lake kind of things. And again, some bigger trees, right? We can see many egrets plus this pelican, all those things sitting together for a long time. Is them also part of this flocking, or they are just simply sitting for?
Yeah. So in literature, this grouping behavior, there is also a definition called aggregation. So a lot of species coming together for a food resource, such as a fruiting tree or so on, that is generally considered different from a flocking. Here, they move in concert. So there is also a movement of each species, a fluid dynamic, very loose dynamics between these species as they move. So at that level, there is a difference. But indeed, these flocks are not just restricted to birds, even though birds are the most studied. There are a lot of flocks within fishes. A lot of studies are going on in those taxas as well.
Yeah. Thanks.
Yeah.
Are there any other questions? Yeah, I just wanted to ask you the initiation of the music flock. It's done by most probably the drongos, right? And they call the other birds to come together. Have you noticed that?
So that is one hypothesis that's there,
Yeah.
and a lot of people have started to look at the propensity of vocalization and how that determines your place in a network. If you create of all the participants in it, does being more vocal mean that you're also more central to that flock? So those questions are being answered. A follow-up study from our NCBS has done a little bit, and it shows it possibly shows that there is a relation there. The more vocal you are, the more likely you are to be central to the flock.
Okay.
Yeah.
Because where I stay now in the city of Cochin, I have racket-tailed drongos in the urban setting, and very often I hear these calls, and they start calling in the morning, and then I hear the calls of the tree pie, then I hear the calls of the jungle babbler. When I go out and see, it's only the racket-tailed drongo. Nobody is there. Probably he's trying to call everyone and trying to tell them, please come and join, but nobody's around to join. Maybe there are
It's a mesmerizing hypothesis even I had, and I had spoken to a lot of people who study vocalization and mimicry in flocks in racket-tailed broncos, but there is still differing opinions. Some say that they do it for the fun of it. The others say that there is a specific role for it. Yeah.
Another thing I've noticed is sometimes these dominant birds like the drongos, they also extract some price from the participants. Like they go and
Yeah.
put piracy.
Very evident with a lot of, they just like play out a false alarm call and then everybody just runs off, and then there is this classic example of the praise coming up and sallying. Yeah. So that is also, we observed quite a lot of instances like that during the data collection.
So thank you. Bird flocks are also called by another term which you are not mentioned, bird waves. And especially when you go into the forest, like what was described by Bates, you can walk miles together without saying anything, and suddenly you have this flock coming. It's like a wave, so, and you don't know what to look at, and they come, they do a lot of antics, and then they just disappear. So that's something which you can experience when you walk in the forest. So I hope this will inspire you people to go out and look at birds in your neighborhood, and when you go for a walk, look out for these flocks. So
And
thank
that is
you very
quite
much.
experiential, as he mentioned, because I met a lot of famous photographer who their still problem is that they haven't managed to take a picture of a flock, because it's an experience. You can't really capture that. You have to see them moving, the sort of relations. That's why I put up just paintings over there, because there are no flocks. Thank you.
So thank you. So with this, we conclude this session. I do hope there are no questions from the earlier for the earlier participants. If you have any questions, maybe we can have 5 minutes more. Would you like to ask questions to any of the other people who presented earlier? Question that you wanted to ask but you did not dare to ask, you can ask now. Okay, then we'll wind up this session. Thank you so much.
Hello. Just assemble for a session photograph. Thank you.
എല്ലാവരോടും ഒന്ന് ഒരു ഗ്രൂപ്പ് ഫോട്ടോയ്ക്ക് എല്ലാവരും ഒന്ന് മുന്നോട്ട് വരണമെന്ന് അഭ്യർത്ഥിക്കുന്നു. ഇന്ത്യയിലെ തന്നെ പ്രമുഖ പക്ഷി നിരീക്ഷകരും കേരളത്തിലെ സീനിയർ പക്ഷി നിരീക്ഷകരും ഒക്കെ ഉള്ളൊരു ഗ്രൂപ്പാണ്. അപ്പോൾ നമുക്കൊരു ഗ്രൂപ്പ് ഫോട്ടോ എടുത്ത ശേഷം പിരിയാം. എല്ലാവരും മുന്നോട്ടേക്ക് വരാൻ അഭ്യർത്ഥിക്കുന്നു.